And thanks for this So thanks for this nice introduction. So as already mentioned, this is a pilot study, and even that it has now nicely explained what the aim is, we are not yet there. So we are still in the phase of processing the data. And also it's a quite large pilot study. So it's not only me working on this project. So it's a whole team behind this. So I'm just a project leader. And that's why I would like to start with the people involved in the project. So it's also Oliver Reutemann and Gabriel Ulrich, which are two doctoral students working on basic research. Then is Yasmin Chan, was a master's student, where I will show some result, what she has produced. Enrique Rayo is the clean lab specialist, which did all the clean lab with the samples from herbaria and collections. Then Nick Zemp is the bioinformatics from the GDC, which helped me with some of the analysis, which you will see already now. And then Andrea Riffel is my lab assistant, and Felix Gugli, Rolf Holderecker, and Alex Widmer are the senior scientists, which are always useful for giving important feedback and having a lot of knowledge about the topic. But with this, I would like to start very basic, and that is, what is genetic diversity in your everyday life? So maybe start thinking about get you in contact with genetic diversity. How does it look like? And so what is genetic diversity? Because I guess that's the most difficult part to convince people why we should monitor genetic diversity, because it's quite difficult. Many people have really big trouble to understand what genetic diversity is. And so with normal audience, I would introduce people and try to see what they think about it, which is difficult by Zoom. So I may start very basic. For me, in the everyday life, that's, as example, genetic diversity. So all this variation you may see in carrots, this phenotypic variation has a genetic basis, and therefore that's genetic diversity. But also dog breeds. I mean, technically seen, these were all wolves, and they have been selected by humans to evolve to different breeds. And all the variety we see here is just because there was genetic diversity in wolves, which could evolve into the different breeds. So for having dogs which are good in looking for sheep, dogs which are able to search for people, and so on. All of this is genetic diversity, and maybe still remain in things we as a human are interested is about food. So all what you can see here is wild mustard, which has been bred and resulting in different food sources for us. And genetic diversity in my everyday life rather looks like this. So I'm a bioinformatician, population geneticist, and that's my everyday life, just looking at 4 letters and how to infer what genetic diversity is. And so why is genetic diversity now important? I mean, we need to be very clear. Genetic diversity is really the raw material for evolution. So that species or population can evolve, there is genetic diversity needed so that they can adapt, as example, to different food source, like the Darwin finch in this case. And it allows species and population to adapt to different environment and to persist in a changing environment. And so therefore, genetic diversity is really crucial for long-term survival of population and species on our Earth, especially given now that we have such strong climate change. And something most people are not aware of is that genetic diversity also contributes to stability of ecosystem. And a nice example are the eelgrass meadows shown here. Only eelgrass meadows which had high genetic diversity were able to cope with increased temperature of the sea. And also eelgrass meadows with higher genetic diversity, they also contained more different species living within these eelgrass meadows. And then, of course, for us again, genetic diversity maintains of ecosystem services. So given all these information, I think genetic diversity per se is considered beneficial and hence worth being preserved and also monitored. But that's where the big problems may start, because there is no systematic monitoring of genetic diversity or has rarely been conducted until now. There is a nice paper from David O'Brien which shows which monitoring currently are running, just to give an overview, and there are only a few. It would be very important to report change in genetic diversity to international authorities or national authorities, but that's still very, very rarely done. And currently now we have COP15 in Montreal, where the new targets for biodiversity are negotiated, and hopefully genetic diversity will be one of those new targets. Now, if we take the focus back onto Switzerland, within Switzerland, we have quite a lot of monitorings going on. But these monitoring are mainly focused on ecosystem or species diversity. So we have a biodiversity monitoring program called BDM. We have the monitoring of common breeding birds, the MHB. We have monitoring the effect of habitat conservation in Switzerland, WBS. We have species and habitat in agriculture areas, so the ALEMA. And you have the Swiss National Forest Inventory, the LFI. So there are 5 different large monitorings really running in Switzerland since up to 20 years, but they are only looking at ecosystems or species diversity. And there is no genetic diversity monitoring at all currently running in Switzerland, or there is one now, which is this pilot study. And so, as mentioned, there is or was in Switzerland no monitoring of genetic diversity running. And so starting such a monitoring project is not a trivial attempt. So that's why we thought we first need to build up the knowledge on how such a monitoring could happen in Switzerland. And so in 2019, we started a feasibility study where we tried to see how could we monitor genetic diversity in Switzerland over a long time period. And we came to the conclusion that we should monitor up to 50 species using, and this would mean about 17,000 individuals which have to be sampled. And the aim would also be that we really have a monitoring, meaning you should have different time points where you have estimates of genetic diversity. And so if you start now, we only would have a single time point, which is not yet the monitoring. So our idea would also be to go back for 10 species, so going back in time using collections, so herbarium samples, and so that we have also time points from the past to see how genetic diversity has already changed over time. And we came to the conclusion that we should use whole genome resequencing data to do such a monitoring so that it is future-proof, because sequencing technologies will improve in the future. It will get cheaper and cheaper. And so a monitoring should be a long-time monitoring, and we should get the best data already now so that we can compare with future data. And the aim would be to repeat this every 5 years. But now, starting with 17,000 individuals and 50 species right now, using whole genome sequencing would be a bit a large burden, and it would be a head start, and it would be very difficult. And that's why we came up to start with a pilot study. But maybe first also we did in this feasibility study, we did a stakeholder analysis to see what currently the stakeholders in Switzerland think about such a monitoring, where they would see the usefulness of a monitoring and where they see some disadvantages or problems with such a monitoring. And we did a network analysis also to understand which stakeholders are important in Switzerland to be included in such a monitoring. And so, as already mentioned, starting with 50 species would really be very difficult. And so we first need to gain some experiences how we can implement such a monitoring on a national scale. So it would be about how do we sample all these different individuals and population in Switzerland that we get good estimates of genetic diversity, and especially also unbiased estimates of genetic diversity, or as unbiased as possible. It's also about the analytical procedure in the lab. So how do we handle all these samples? How do we process them as efficient as possible? It's about also developing the bioinformatic pipelines, so to be able to process efficiently the genome-wide data. And of course, such a pilot study already will give you a first baseline of genetic diversity of these of some species. And with going back in time with museum samples, such a monitoring would also help you to get really the first changes in genetic diversity over time. And of course, it's important, again, such monitoring should not just be done because we can do it, but these results should go into real measures, and therefore the cooperation and including of stakeholders is very important. So we are in close collaboration with as many stakeholders as possible to really make best use of this genetic data. So this whole project is the Swiss project is financed by the Federal Office for the Environment, and because they are financing most of the monitorings running in Switzerland. So maybe coming more to what we are really doing in this monitoring. So it's a 3 years pilot study. And we decided to start with 5 species. And these species have been selected because they live in habitat of national importance, or they are in landscapes which are anthropogenically modified. So what you're looking at as example is the hairtails cottongrass, which is a species living in peat bogs. We have the Natterjack toad, which lives in amphibian sanctuaries. We have the Carthusian pink, which is a plant living in dry meadows. We have the false Hess fritillary butterfly, which is a butterfly species that lives in edge habitats. And we have the Yellowhammer, a bird species that lives in agricultural areas, which is, of course, highly modified by humans. So the aim would be to sample about 150 populations. It means 30 population per species and up to 1,000 individuals. And given the experience we got from the sampling or selecting these 5 species, we were also involved in a IUCN guidance document on how to select species. So those interested, Christina Wilson has wrote up a nice guidance document, how you should select species for such a monitoring project. And for 2 of the species, especially for the butterfly and for the cottongrass, we all try to go back in time and getting samples, or 400 samples in total of both species from collections or herbaria. And as already mentioned, we're really aiming for whole genome resequencing data, and also to really get good estimates, we aim for assemble all the 5 species de novo using PacBio and HiFi data. And the aim is to sequence all these 1,200 individuals with about the 10x coverage. As mentioned, the 10x coverage whole genome resequencing is really that we get accurate estimates and which are also future-proof, that we can compare it with future data. So related to that, we also had a meeting to figure out what are ideal indicators which help to report changes of genetic diversity to national or international authorities. And we came up with 5 meaningful and intuitive indicators, or hopefully they are intuitive. So the first one is, of course, about genetic diversity per se, and we came up with Waters and Theta, because Waters and Theta is one of the genetic estimators which change or which reacts the fastest to changes in genetic diversity. Then we're aiming for looking at inbreeding, and there we thought, okay, rounds of homozygosity, because we have whole genome resequencing data, may be a very, very powerful approach. Then genetic connectivity, that's still an indicator we need to see which is the best estimate to use. So currently we will, or we in the future will test bias, but we have not yet started with that. And then Oliver Reutemann is developing a new quantitative approach to figure out the adaptive potential of these population and species, but all of this is still work in progress. And then, of course, one of the well-known estimators would be NE, so the effective population size, to understand future effects of drift to these populations. And these 5 indicators where we start and try to get first values for. And of course, a real monitoring would mean we would like to see changes of these indicators over time, and not only getting point estimates. Currently, we're only getting point estimates, except for the 2 species where we go back in time. And now here I need to be honest to you. So currently, we more or less have only data about genetic diversity. All the other indicators, we are still working on the data or have not even started, because we are really still in the middle of the whole project. So, of course, the first step in such a monitoring program is to figure out how should we sample these different individuals, populations, and species across Switzerland. And we came up with a proportional stratified random sampling strategy to sample this. And I show you how we have developed this sampling strategy at the example of Dianthus carthusianorum, so the Carthusian pink. So first of all, we get occurrence data of all the species. And then we restricted the occurrence data to data points only starting in 2000, so that we have more recent location where the species still occur. And we got maps like this where these species have been recorded. As you can see on this grid, there are hotspots where a lot of recording have been made, but this is a strong bias because that's maybe areas just where some good botanists are. So that's why we overlay the whole Switzerland with a 5 by 5 kilometer grid to see in which of these 5 by 5 kilometer grids Dianthus has been observed. So this can be areas where you have a lot of observations or other areas where you have only very few observations. So this ends up with a 5 by 5 kilometer grid in pink where the species can occur. And then from these grids, where there is occurrence data, we randomly select 30 grid cells. And additional to just randomly select them, because the Federal Office for the Environment would like to know changes across also the 6 biogeographic regions, which are here colored in different colors. So we had to do a stratified random sampling, meaning given the amount of square we have been observed in each of these biogeographic regions, we sample proportionate to those. So you see if we have 97 squares, so on average from these 30 random squares, 5 should come from this biogeographic region here in the north. And so finally, we got our 30 random grid cells, and that's the place where we will send out, or we have sent out our helpers to get these samples. And of course, because this is a random sampling, some of these places, maybe it's very unlikely that these field helpers will find the species. We have not only selected 30 random grid cells, but 40, so that we have replacement grids if they couldn't find the species, because they may went extinct in the meantime, or they couldn't find them. And that's the result. And to be honest, I'm quite proud about that we reached this more or less within a year. So the main sampling season was in the year 2021. And we had 29 people in the field which tried to sample these 5 species. And they were able to sample 158 populations across all 6 biogeographic regions. And they sampled more than 1,200 individuals, which I think is an amazing result. Even given that the spring and summer 2021 was in terms of weather very bad, it was very wet, it was very cold. So especially for butterflies or toads, it was very difficult because the breeding season and the flying time was massively reduced, and we had quite some trouble, especially for the butterfly. In eastern Switzerland, we had to do some resampling in 2022 because we couldn't find the butterflies in 2021. So maybe I should also refer to which dot represents what. So orange dots are Melitea diamina, the butterfly. Yellow dots is the yellowhammer, so the bird. Brown dots is the toad, Epidalea calamita. Pink is Dianthus carthusianorum, and green is Eriophorum vaginatum. These are 158 populations we have sampled. So now we also would need to try to validate whether this random sampling was really successful. And to do this, we estimated the climate space of each of these species. So we compared the mean annual temperature to the annual precipitation. Each gray dot represents all the observations that have been made in Switzerland for this species, and the red dots are the population we have sampled. And you can see that the climate space are all the gray dots of this species, and the red dots are those which we have sampled, and they are, I think that our sampling really shows that we have a nice random sampling, not only on the geographic scale, but we also have a nice random sampling in the climate space of this species. And this is true for all 5 species and all the 4 other species. And what is also obvious is, as example, that the toad has a very narrow climate space. So it only lives in rather warm and more or less a bit drier areas in Switzerland. So a next step, of course, is that you can work with whole genome data. You need to have a reference genome, and none of the 5 species until now had an annotated reference genome. So we had to assemble them on our own. And we used PacBio Hi-Fi, and for some of the species also OmniC, which is a specific case of Hi-C data to assemble these genomes from the fragmented pieces we get to full genomes at the end. And having high quality reference genome is really important because, first of all, it increases the accuracy of your different estimators you will calculate, but especially it reduces the computation time. So if your reference is in bad quality, meaning in many contigs and has a lot of errors, the calculation time can really explode, and it will take forever until you have done all your mapping, all your SNP calling. So having good reference genome is really highly beneficial. And nowadays, with these new technologies, it's really even feasible, which I would like to show you in this case. So we assembled genomes from 446 million base pair up to 3.8 billion base pairs, which is the toad. So the toad genome is even larger than the human genome. And we were able to really, or nearly or closely assemble these genomes to reference genome to chromosome level. So in bold here mentioned, these are the big contigs we were able to assemble, and behind are the numbers of contigs in total we had. And so most of the genomes are really very close to chromosome level assemblies. And the BUSCO score is some sort of the accuracy of these genomes, because BUSCO, this is a list of several thousand conserved genes, and meaning in this case for Eriophorum, 98.4% of these conserved genes we could find in our assembly. So indicating that all these genomes, even that we started from scratch, are rather high quality. Then as a next step, we have sequenced all these individual using whole genome resequencing, using an Illumina machine, and the aim was reaching up to about 10x coverage. And this is just showing you how this would look like. This is the example of Melitea diamina when we map one individual back to our reference genome. So you can see that this is the entire genome, all the different chromosomes. This arrow indicates 597 of the 603 megabases, which are the 34 large contigs. And only this one here are the remaining few contigs of lower quality. You see no reads are really mapping on this. You can see on average, we have about a 15x coverage for each position along the chromosome. But most of you probably have spotted that there is here and here a massive drop where we go down to about a 5x coverage. And this just indicates that our reference assembly is really good, because these are the two sex chromosomes. And Melitea is a rather young sex system. So we have a ZW system, and this is the Z and the W chromosome of the species. So we were even able to assemble the very difficult to assemble sex chromosomes. And whenever you work with whole genome data, there are many unexpected surprises to come, like being able to detect these sex chromosomes and many more. So I'm looking forward. So we're still doing all the analysis at the moment. Maybe now would I really like to come to some of the first results, and I really need to be clear here. We just got the data in fall this year. Most of the data, or some of the data still is coming in. So we have not yet finalized all the sequencing, but we're close to it. So all the results I'm now showing is really either a subset of the data sets or only freshly analyzed. So be careful with the interpretation. They can change. Maybe I would like to show you why we should go for whole genome resequencing data. This is a PCA, which shows the genetic structure of the 30 population of the cottongrass. And you can see we can more or less nicely resolve these population when you use only the first chromosome, which is about 100,000 SNPs. But if we now take the entire genome, and I now also turn this figure that it's comparable, you see that more or less the structure remains the same. So we have these yellow and violet population here, the blue and violet populations here. So we resolve the same, more or less same structure, also using the whole genome data, but we have much more resolution. We now really can separate each population very accurately using whole genome data, which was not possible if we only used 100,000 SNPs. So going for a whole genome resequencing approach really increases the power massively. And as already shown here, we're talking here about 100,000 SNPs, which is already a lot, but going for 1 million SNPs is even a more boost of resolution. So if we now really dig into genetic diversity, what I present you here is now really the baseline of genetic diversity. This is not really a change in genetic diversity, but that's the current status of genetic diversity of the cottongrass in Switzerland. And these are the 30 population we have sampled. And here it's the only and first species we have started calculating Waters and theta, and everyone that knows population genetics knows that these values are not really Waters and theta values. This is Waters and theta per site, because the big problem working with such large genome is estimating real Waters and theta. You would need to calculate all invariant sites, which is computationally extremely intense. And we're still working on a way to surround this to really get real Waters and theta estimates. But it still means that in this case, green are population with high genetic diversity, and red are population with lower genetic diversity. And in this case, it's nearly half the genetic diversity of the red population compared to the green populations. And there are some clear patterns, and it seems that there is a hotspot in central Switzerland, and to the border to the north, there are a slightly lower genetic diversity populations. And what is also interesting is we calculate the observed heterozygosity of each individual. So each bar plot represents a population, and each dot is one individual within this population. You can see that there are populations which have rather high genetic diversity, and all individuals within this population have rather high genetic diversity. And if there are populations with lower genetic diversity, that the spread of the individual genetic diversity is also much larger, which maybe is already an indication that these populations suffer from inbreeding, that these individuals here down really have problems with inbreeding. But that's something we are just digging into with analyzing runs of homozygosity. So we also can try to visualize genetic structure of these populations using the PCA. So this is an approach where each corner in the PCA get a separate color, and then each population get an average color to what it belongs to. And so the colors of each of these populations can then be transferred on the map. And so looking now at the cottongrass, you see that there are more or less 3 different clades, or we may be evolutionary lineages that we can observe in Switzerland. So we have one in eastern Switzerland, which is quite distinct. And here in the valley, we have also 2 populations which are genetically quite distinct. So these are things we need to take in consideration, because if I'm going back, you also see that it are these populations which have a slightly lower genetic diversity. And this lower genetic diversity may not be because of human action. This genetic diversity difference may be just because of evolutionary lineages. Then I would like to come to the second species where we have analyzed the entire data already, or not analyzed, but at least called SNPs of the entire whole genome data, which is the Yellowhammer. And in this case, we have 6 times more SNPs. We have more than 6.2 million SNPs called. And this is again the map of genetic diversity, but now it's only averaged observed heterozygosity per population, not yet Waters and theta, because I could not yet calculate that. And now we need to be very careful because the difference here is, or the difference in observed heterozygosity ranges only from 0.6 to 0.2, from 0.26 to 0.25. So the genetic diversity differences between these populations there is nearly no difference, but still we have colored them from green to red, which is of course a bit misleading. This just shows that at least for a very mobile species like the Yellowhammer, genetic diversity is still rather high in Switzerland, or high, or rather similar for all the populations. However, if we now look within each population, you see that there are populations where all individuals have very similar genetic diversity, but there are also populations where genetic diversity is highly variable between the different individuals. And those which show such large patterns of variation of genetic diversity within each population are also those which have a bit lower genetic diversity. And this may already also be some signs of isolation or maybe inbreeding. Because if we compare this to the occurrence data of the Yellowhammer, or the change of occurrence since from 1996 to 2016, we see there are regions in Switzerland where the occurrence had, they lost some of the bird populations, and this will now be interesting to really compare this 1-to-1, but we are not yet there. But these are steps, the next steps we would like to address. Then I would like to come to the amphibian with the biggest genome we are looking at, which is the Natterjack toad. So the genome is 3.8 billion base pair, which means larger than human genomes. And so sequencing these individuals is quite costly. And given the bad weather we had, there was a big problem that the breeding between common toad and Natterjack toad were for once at the same time. And we were not sure whether there was some sampling because we sampled tadpoles and eggs. And they can be it's very difficult to distinguish them. So we did a barcoding before we sequenced these individuals. And out of the 144 individuals, we identified 5 common toads and 1 edible frog, which we have excluded from the sequencing, so to save a little bit of money. And here now, you need to be careful. So this is just a subset of 2 contigs. It's only 30,000 SNPs. However, the results are here already very interesting. So again, these are the observed heterozygosity, so genetic diversity of these toads across Switzerland. Red again indicates rather lower genetic diversity, green in this case higher genetic diversity. And here for the toad, we have really massive variation in genetic diversity across Switzerland. So again, there's a twofold difference in the levels of genetic diversity. And it is even stronger if we look at individual genetic diversity. See, these bars or boxes are really wide for all the different population, which indicates that maybe in some location we have already problems of inbreeding, but that the runs of homozygosity will give us more information about this. But we have not yet done the analysis because we have not yet called all the SNPs for these large genomes, as this takes several weeks to do so. Then also genetic structure still is quite interesting, even working on such a small subset. We see that there is closely related individuals to Western Switzerland and another clade to Eastern Switzerland. And in between, there's maybe a third clade and a lot of interbreeding probably going on in this area, which is also in line with genetic diversity. We have among these region here the highest genetic diversity of the toads. So if we take all of this together, I think it clearly shows how much more information we gain with using genetic data compared to just observation data. So this map here shows you the red dots are the observation in the last 20 years of the Natterjack toad, and green areas is where it's expected that the toad still exists, whereas the red areas are areas where the toad went extinct in the last 20 years. And of course, already this gives quite a lot of information, but I guess the genetic data already shows you now clearly that there are populations where genetic diversity is much lower, and therefore they're probably more endangered. And it also shows breakpoints of connectivity between these populations. So genetic data, from my point of view, really gives much more or already more information than just occurrence data, which we had until now. And then just a very complex figure. These are the pairwise FSTs between all the populations. The only thing you need to focus on is on the FST scale here. And you can see that dark red means an FST of 0.6. And that's for FST a really high value, meaning these populations here are very distinct compared to other populations. Either indicating, again, evolution lineages or already very strong separation of these toads from each other, which would be worrying. But this needs more work to dig into to know whether this is human-made, this strong differentiation, or whether these are evolutionary lineages just from the last ice age. Then coming to another highly interesting result is from the Carthusian pink, because if we first look at this map here of genetic diversity, in this case, it's again a subset, a very small one of only 14,000 SNPs. You will see later why this SNP set is currently that small. You see again that you have quite strong variation in genetic diversity in Switzerland, which is quite mosaic-like. And also, if you look at the genetic structure, we find about 4 different clades. So the blue one, the pink one, the brown one, and the green one. And you see suddenly this brown one are found in the blue one, which makes not a lot of sense and started us to puzzle. And the other thing you observe is that the amount of variation explained in this PCA on the PCA axis 1 is really high for genetic data. So we are here at nearly 25%. Before we were always in a range of about 3 to 5%. indicating that very strong genetic structure in Dianthus in Switzerland, and that puzzled us, so that we thought it's time that the master students dig into this a bit more and opens the scope from Switzerland to the entire distribution range. So we got samples, there are 254 samples of Dianthus carthusianorum from more or less its entire distribution range. And we even included 3 outgroups so that we can have a proper phylogenetic tree and understand these evolution lineages of Dianthus better in Switzerland and across Europe. And now, to save some money, we sequenced the samples outside of Switzerland with DDRAT markers, a reduced representation sequencing approach. And we developed a new approach so that we can combine DDRAT data with whole genome resequencing data by producing a SNP panel that is based on the DDRAT data. And as mentioned, this is the master thesis of Yasmin Chan, which has been defended just a few weeks ago. And so doing now with this data, a phylogenetic tree using RuxML, she could identify that across Europe, we have about 6 different evolutionary lineages of Dianthus carthusianorum in Switzerland. So we have the Western European lineage, we have the Italian lineage, we have the Western Austria or West Austrian lineage, the Eastern Austrian lineage, we have a lineage in the Czech Republic, and we have 1 lineage in Romania, which are clearly and highly supported by the phylogenetic tree. So indicating that in Switzerland, we should expect to have 3 different lineages. And all these lineages, that's now also quite important to realize, they have different genetic background because they have different evolutionary history, because their refugia were large or smaller, there were more interaction, hybridization in the past going on. And it's clearly to see that especially the Italian lineage and the Romanian lineage have lower genetic diversity compared to the 4 other lineages. And if we now dig into Switzerland, surprisingly, we find 4 different evolution lineages. So we have the yellow one, the blue one, the red one, but we also have the violet one from Romania. And this is, of course, now slightly puzzling. And to dig into more to that, Jasmine looked at the maps and all the positions where these populations have been sampled and sequenced. And for all those which are not connected by a polygon, so all these spots here, she has realized that these were populations from places where in the last 10 to 15 years there were renaturation happening, and this means this was artificial seeding. And also from the genetic data, we could identify that these populations, all of those here, were artificial seedings. And this means from all our samples we looked at, 37% were artificially seeded, which was much higher than we expected. We knew that Dianthus is strongly artificially seeded in Switzerland, but we did not expect such clear and detailed results. And for Switzerland, this means 4 populations, this one here, this and this one here, they have been seeded with seed material from the wrong evolutionary lineages, which can be problematic because these populations, especially the violet ones here, they are from Romania, so really far away, and these plants may be differently adapted to different environment, and also, of course, would allow, or it would happen that there will be in the future gene flow between these different lineages, and that's why monitoring this artificial seeding and seeing the consequences of such artificial seeding is very important. With this, I come to the last species we are monitoring, and I just realized time is running. So this is now the false Hess fritillaria, the butterfly, and there we are really just starting analyzing data. So we still miss all the samples here from Eastern Switzerland because we had to sample them this year, and they are just now being sequenced. But this is just a subset now of 186 individuals and 66,000 SNPs. So also, again, only a few SNPs and not the entire genome. But again, we can see quite some hotspot rather of genetic diversity and areas where genetic diversity is a bit lower. And maybe more interesting is also the genetic structure reflected by the PCA. It seems that we have clear patterns of isolation by distance between these butterflies, and I'm really looking forward to fill this gap here to understand better how these different populations are connected. and how they interact with each other. But that's still working in progress, and we wait for the new data. So what I have shown you now are more or less all just, or not all, they are all just one time point estimate of genetic diversity, which we observe now. But the monitoring would mean we should characterize and quantify the change in genetic diversity over time. And a way to do that is going back in time, as Van der Valk has nicely done this. So they got gorilla samples about 100 years ago and compared it to the genetic diversity from nowadays. They did it for the Grau's gorilla as well as for the mountain gorillas. And interestingly, they only found in the Grau's gorillas, or the flatland gorillas, a strong decrease of genetic diversity within the last 100 years by using DNA from collections. And we are aiming for something similar. So we would like to quantify change in genetic diversity in two species, so in the butterfly and the cottongrass. And we got 202 Melitea, so butterfly samples from collections, and 226 samples from Eriophorum vaginatum, so the cottongrass, which we are currently sequencing. And these samples are somewhere between 70 to 218 years in age, and are therefore also difficult to handle. So all these samples need to be processed in clean labs, which is highly challenging and much more work than I expected. To dig into this a bit in more detail, so we went to all the herbaria in Switzerland. As example, for the cottongrass, we found nearly 700 herbarium samples. And all of these 700 herbarium samples were then digitized and georeferenced, meaning we were reading, or some colleagues were reading these labels and try to find on old maps where these Eriophorum samples have been sampled, and then also compare to nowadays maps and seeing, okay, these habitats nowadays, as example, got completely lost. And we did the same also for the Melitea samples, where we found 1,700 Melitea samples in these collections. And all of this was done with Digitalis, a database tool available from our collections. And so for the hair's-tail cottongrass, we aimed for an individual based approach. So we got 226 samples distributed across Switzerland. We tried to really spread them as much as possible. And Gabriel Ulrich, he now, or this summer, went to the field and visited all these sites again to see whether Eriophorum still is there, and he sampled contemporary sample if they were present. And interestingly is that at 37% of the 226 locations, he could still find Eriophorum. These are all the green dots, whereas the yellow dots represent sites where he couldn't find any more Eriophorum vaginatum. But if we now focus on the central plateau, this is this area here, which is the most or strongest populated area in Switzerland, where a lot of wet habitats have been dried for agriculture or buildings. So there we lost much more of the Eriophorum population compared to the rest of Switzerland. And if we plot this now again on the climate map, you see that we have especially lost populations or individuals which live more in warmer areas and maybe even a bit drier areas. So the big question now is, did we already lost adaptive genetic diversity, or that is linked to rather warmer and drier habitats? So the climate that, given the climate change we are facing, probably will be more prevalent. So that's one of the big questions we would like to address. Just to show you that working with herbarium samples is quite tricky. So sequencing historic DNA has a big problem that you have DNA damage in terms of very short DNA fragments. In our case, it's only about 40 base pairs. You have the deamination of the ends of the reads, so the C to T transformation at broken DNA sites. And also you have, of course, lower endogenous gene content because there was mold and bacteria and other things growing on these plants, which you also sequence at the same time. And so for 2/3 of all the samples, the endogenous gene content is higher than 40%, and that's the samples we can work with. And also still that these fragments are very short, First results really show that we nicely can map them on our reference genome. So that's a mapping with contemporary data. You see the mapping quality is very high, about mapping quality of 60, and we get the same mapping quality also for the historic DNA. So there's a lot of work included to do working with historic data, but it pays off and it works. But you need to be clear that you need to sequence 6 to 10 times more for historic samples compared to contemporary samples. So it's really expensive and time consuming. With this, I would like to give you also a short overview for the butterfly. So there we went for not individual-based sampling, but a population-based approach. So we used 3 different time horizons. So one from 1900 to 1920, one from 1960 to 1980, and of course the contemporary samples. And we tried to have between 3 to 10 individuals in each of these time horizon. And this allows us to have comparison from different time horizons, or from the far back in the past to recent, or from more recent to contemporary samples. And overall, this allows us, with the sampling design we came up, to have 25 population comparisons to see how genetic diversity has changed over time, over different time periods in Switzerland. And this is a bit shown on the map. Of course, the big problem with historic sample is that it's very difficult to find samples at the same position nowadays. So green is the contemporary sample as example, and blue is one of the younger historic sample, or here's a triangle, the contemporary sample, the old sample, and the young sample. These are these population-based sampling we try to infer from. And additionally, we also sampled or sequenced 10 extinct population, where nowadays we can't find any Melitaea anymore. So maybe a last word to working with historic DNA. You have to work in clean rooms because of contamination and the low endogenous gene content. And it's much more time consuming than I expected. So Enrico Raya was for more than a year now in the clean lab to really do all this work for 400 samples only. However, I mean, he did an amazing job. So we got enough DNA from a single leg of a butterfly. So this means we really can work with samples from collection. We only need to ask for 1 leg, 1 out of 6 legs. This is enough to really do this work. So with this, I really come to my 2 last slides, a bit over time, but just to sum up. So it was a pilot study to see whether monitoring is feasible. And I would say, yeah, it is feasible. We came up with an ideal sampling strategy for Switzerland. The sampling success is strongly weather dependent. That's what we had to learn the hard way. And I hopefully could show you that genomic data really increases the power and the resolution massively. And that we gained now a baseline of genetic diversity for 5 species, but the real monitoring would mean you should be able to see changes of genetic diversity over time. And museomics is a way to reach this goal much faster than just waiting the next 5-10 years until we have the next monitoring rounds happened. What are the benefits of such a monitoring of genetic diversity? You get an early warning system to see whether we have lost connectivity between populations, whether we have regions with lower genetic diversity. You may also discover effects of artificial greening or wrong seed sets used. And you can detect changes in genetic diversity over time. And this allows you to have some specific measures, conservation measures. So you can plan target connectivity measures between some of these populations. You can consider evolutionary or locally adapted lineages for translocation or artificial seedings, which from my point of view is what we can already reach with the data that we have now. But I also would like to be clear that what I have presented here is the Swiss model, which is a luxury model going for full genome sequencing and so on. And David O'Brien has made a nice paper about showing what other monitorings are currently running. And I think we need to be clear that we should have a minimum set of indicators that all countries can calculate with the available data. Therefore, I clearly advocate also for the approach that John Hoban proposed, that we should have very simple indicators like NE from NE estimate for each population, the proportion of population which are maintained or getting lost, and maybe just very simple, how many monitoring of genetic or how many genetic data are available for each species and country, so that we can compare our results with the different countries and really start monitoring on a very simple way changes in genetic diversity. So I think, of course, the Swiss model is very useful, a lot of fun to work on, but for most of the countries, we should see a rather more simple approach. So with this, I'm at the end of my presentation. I would like to thank the Federal Office for the Environment for financial support, as well ETH Zurich, the Foundation Geobotanical Rübel, Research Institute Rübel, for technical support, Genetic Diversity Center, the Functional Genomics Center Zurich, and those which like to get even more information, visit our homepage or follow me on Twitter or write me emails. So thanks a lot, and sorry for being a bit over time, but we had a bit of chat at the beginning. So